On this podcast, you'll find interviews with high-performing, successful individuals in life sciences. On a weekly basis, we cover their proven methods, principles, strategies, and mindsets to implement new technologies that scale to meet the needs of people in our world.
Hello and welcome to Life Science Success Podcast. For those of you who don't know me, my name is Don and I'm a consultant in life sciences. I help companies manage complexity and increase performance. Today I'm joined by Kyle Giffin. Kyle is one of the founders of LatchBio, and they're currently working on building a platform to disseminate the data and infrastructure for the biocomputing revolution. Welcome, Kyle.
Thank you so much, Don, for having me.
Yeah, sure. So can you tell the audience just a little bit about yourself?
Yeah, absolutely. So my name's Kyle. Nice to meet you, audience. I'm one of the co-founders of LatchBio, and before that, I studied neuroscience and data science at UC Berkeley. And yeah, happy to get in a little bit deeper into the story here of how Latch came to be, but it really started with a convergence of personal interest in the life sciences, neuroscience, how the body, how the brain work, and then combining that with building really, really good technology to help scientists, which has always been something that I've cared deeply about.
Now, there are three founders of the company. Is that correct?
Yes. Yeah. None of this would be possible without the two co-founders and amazing, actually two of my best friends in the world that I started this with.
And how did you guys just meet at UC Berkeley, or was it something that you knew them before even you went to UC Berkeley?
It's funny, we met at UC Berkeley, and an unlikely origin story. We actually met at a fraternity freshman year at UC Berkeley, um, and it was interesting because we had met there, but our interests, the three of us, had a lot more of like the intellectual side of being really curious and like wanting to dive more into the science, wanting to build great things. So we slowly started just having a lot of conversations and getting together outside of that environment. And then, I mean, through actually thousands of hours of conversation, realized that we wanted to work together on something. And at the time, we had been building all sorts of things. I mean, you can imagine we're sophomores at UC Berkeley. So we're 20 years old, 19 years old. And outside of class, we're just like, we want to build software together. We want to build things that are useful for people. What can we do? And very quickly, we started building different cool little software apps that weren't actually gaining a lot of traction. A lot of people weren't using them, but it gave us the experience to know we can actually build something that someone will look at and potentially try and use. And even that alone was exciting to us. And so from that moment, we actually stopped building things and we said, okay, well, what do we actually want to spend our lives on? Because we've been building together for a few months, but what do we want to spend 10 years on that will actually have a large impact on the world? And that's when we started to focus a bit more on what problems we care about. And it's an interesting thing to ask yourself, what problems do you care the most about in the world that you would want to spend a decade or 2 decades actually trying to solve? It's kind of a philosophical question. And so we talked for many hours about that and landed on something involving helping people at a very large scale and then building software, because that was our sort of background, and what we knew we wanted to do was build software to try and do that. And so a pretty orthogonal way into biology than I would say most teams. But long story short, my CTO and co-founder Kenny had many years of wet lab research experience since he was 14 years old in the lab, pipetting, doing directed evolution, eventually at Berkeley Lab, and then working at Serottany, which is a cell therapy startup in San Francisco. And basically what happened was we started asking about the work he was doing at Serottany. And as an 18-year-old, he was building all their software infrastructure as an intern, getting paid $15 an hour to build this extremely complicated software stack for a bio company. And we started looking more at this and we said, I wonder if this is happening at other companies. And through a series of hundreds of interviews of biotech companies in the Bay Area, Boston, Germany, UK, across the world, we learned that every biotech company is reinventing the wheel, building their own software platforms in-house to store and manage these massive volumes of data that are being generated from sequencing, microscopy, and other high throughput research methods.
Yeah, it's funny that you say that because I mean, I guess there's two things. One, so in some of my graduate and undergraduate work, I mean, there were different opportunities to sort of work on different businesses. And I mean, primarily the biggest one that had the most traction and probably made it furthest down the runway was starting a brewery. So to come here and focus on a bioinformatics platform is totally, I mean, a bit of a different scale on your side. So
to be candid, I can relate a little to that. We actually built a dating app when we were sophomores. That was one of the first things we built before we ended up going into bioinformatics, biology. So yeah,
there you go.
You don't always start with the most bespoke thing.
Yeah. So the other question I had for you regarding the, kind of the overall journey, because you touched on it with regards to Kenny. So as you kind of looked at all of the biotechs and things like that and started to collect more and more information about, what you might, form Latch to be, did it take much to sort of align around that, or was everybody just, at a point where you just said, hey, look, this seems like a really good idea and it would definitely have a big benefit on the world?
Yeah, it's a good question. I think for us, we really didn't even know what the idea was. All we knew was that we would talk to scientists and we would talk to bioinformaticians and ask them about their problems and ask them about how they were wasting the most time every day and the things that frustrated them and annoyed them in the lab. And we got hundreds of hours of responses. And it just became very clear that no great software tools have really been built to address the needs of these scientists. They were using Excel spreadsheets for everything, physical hard drives, local servers, sometimes pieces of paper to track things. A lot of really, really outdated tools, and none of them had caught up to, the environment you now have with a modern molecular biologist is they're running sequencing assays like whole genome sequencing, RNA sequencing, whole exome sequencing. They're looking at individual single cells and DNA expression on those single cells. So it's not uncommon for one biologist to generate hundreds of gigabytes of data from a given experiment. And now you imagine managing dozens of biologists, tens of experiments, and you really need someone with the expertise in computer science, software engineering, cloud infrastructure to help these scientists. So that's what's happening today. And we really didn't align about an idea. We just aligned about this problem. Why is this happening? And I'll illustrate it with one more example, which is that if you're a team of 15 or 20 biologists, you'll be lucky to have one bioinformatician. So all the data generated from those 15 people is sent to that one bioinformatician who's scrambling to process everything locally on their computer to set up infrastructure, cloud instances, databases, versioning, and they're scrambling to basically build a software stack that can support all these scientists. So it really just doesn't make sense. It's a multi-year engineering effort, multiple millions of dollars for every biotech company, whereas you could just have one team of engineers focused on building the best platform for many biotech companies that they can then plug into. And the last thing I'll say is I really don't think it was clear to us that solution was the solution until 4 or 5 months of interviewing these scientists. So it took a long time to arrive at that.
Yeah, I'll bet. I mean, I'm kind of smiling and nodding because I lived through some of this today with some of my clients. Some of my previous employers, a similar sort of thing. They all built their bioinformatics pipeline in-house. They all more or less are living with it. And then, you have things like the pandemic that more or less cause people to kind of question, where is all my data? How do I take what I have and kind of, leverage the things that I do have as well. And I know there were a lot of larger sort of corporations looking at, how do we create a bigger data catalog? How do we have more and more information and access to information as well? And it's so critical today. You're just being able to reuse and use the data that you have as much as possible.
Yeah. And the ironic thing is that the companies that are the best at this are companies like Google, Netflix, Facebook, where they've assembled the smartest engineers, polymathic people in the world with backgrounds in math, theoretical computer science, software engineering. And they're spending tens of billions of dollars to build these crazy data platforms to optimize advertisements. And that's what my co-founder Alfredo was doing. Actually, he worked at Google and Facebook before we started this company together. He was building these systems and working with people. And then you would compare these incredible systems to optimize ads versus what the modern lab has that's trying to cure cancer, or trying to address aging, or cure this rare genetic disease that affects 10,000 infants a year, where their life expectancy is something like 2 years old. And they're just using thumb drives and local scripts. So it just doesn't make sense. And I think a lot of the problem is that the great talent in the world, the great engineers, are going off to these big tech companies instead of joining bio and biotech. And I think that's changing over the last couple of years, especially with the changes that have been brought on by the pandemic.
Yeah. So tell me a little bit more about what is LatchBio and how is Latch different?
Great question. So I would say at the base level, what LatchBio is a cloud platform for you to store large amounts of biological sequence data, for you to analyze that data and process it, and then for you to visualize the results all in one sort of flow, all in one stream. And it's accessible both by the wet lab scientists who are normally pipetting at the bench, and they have their laptop out next to them on their bench. And it's accessible by them through a very easy to use GUI, but it's also accessible by the bioinformaticians, who are the data engineers. They're the ones actually writing the command line like scripts and logic to take in raw data and process that data into something useful. So a few examples of like the flow that Latch supports, right? You mentioned COVID. One of the labs we work with processes thousands of clinical COVID samples every month to tell patients in the Bay Area what variant of COVID they have and whether or not they tested positive. So here's the flow, right? They go out, they have all these health clinics, and they test patients. They gather the DNA samples from the nasal swabs from the patients, and then they take in those samples and they put them in a sequencer. They sequence the DNA of each patient, and then they generate a bunch of files. These are called FASTQ files, and they just contain raw sequence data. The scientists at that lab, it's the Innovative Genomics Institute at Berkeley, will drag and drop those files into Latch. She can store all of them in Latch. She can manage and name all her experiments in Latch. And then she clicks on a SARS-CoV-2 pipeline. She clicks on it. She drags and drops those files into the pipeline, and she launches the pipeline. And what that does is it does a lineage and a clade analysis on the samples, and it calls those samples for variants and returns to her a beautiful CSV that says, here is every patient's sample, and here is whether or not they have COVID-19 and which variant they have. And then she takes that CSV and she sends it back to the health clinics, and it tells the health clinics, it gives the results. And that would be the flow for one scientist at one lab, but you can imagine this being used for the same exact flow for RNA sequencing or whole genome analysis at many other labs.
Yeah, it's one of the things I've had actual previous podcast guests on as well. I mean, I know that a lot of people are working on, liquid biopsy and things like that, and it just feels like we're so far away from that. But again, the main reason why I believe is because everybody's approaching each and every pipeline differently, and the base science is different as well. So it's kind of, you have the worst of two sort of situations, right? That, one, your sort of base science and the ability to sort of replicate your science is a little bit more challenging. In addition to that, the pipelines are definitely different. So this is where I, whenever I look at Latch, it's like, honestly, this might standardize some things that, were variables before.
Interesting. Yeah, I'm curious also, I definitely have thoughts there. I'm curious also the context in which you encountered this. Was this at, what kind of data were you dealing with where you noticed this?
It was all cancer data. It was cancer data. So most of the companies that are working on liquid biopsy today are primarily focusing in on ways to sort of do one cancer test for all, which if you know much about cancer, it's pretty variable in itself. But on top of that, you kind of, look at the fact that you have, the approach in terms of the way that you're going to analyze the data being, so vastly different. It's a bit of a challenge as well.
Totally. So thinking about this case, this is an interesting way to illustrate how do you trace the versions of every new pipeline, and how do you accommodate all the new logic that's being written by all the bioinformaticians? You can't standardize that. And that's a huge objection to Latch, is how do you create standard pipelines, right? So what we've tried to do here, and it's a very hard problem to tackle, is to allow bioinformaticians who are writing pipelines to develop their pipelines on the platform. So are you familiar with GitHub, or have you used GitHub before? Do you know what that is?
I'm familiar. Yeah.
Familiar, right?
And,
so it's a place
and I
where
think most of the audience would be, but maybe you want to kind of touch on what it is.
Sure, yeah, totally. So GitHub is basically a place where developers who write code for any kind of reason will upload that code, and anyone else in the world can access their code and reproduce it and use it. So it's like the de facto place in the world where if you're publishing your code anywhere and you want other people to use it and see it and fork it, and do their own fun things with it, you use GitHub. The benefits here are that every piece of code is versioned, it's super traceable, it's super reproducible, and it's publicly accessible. It's open source, so other people can access it. Now, in bioinformatics, what happens is a lot of these tools are published in papers where the person who wrote the tool didn't make it reproducible. They didn't publish a good repository. There's no test data associated with the pipeline, so you can't really test it out. And maybe it's written on some outdated, it's written in Bash or like some outdated version of Python.
Right.
So it's really hard to set up. You have scientists like scratching their heads, spending 12 or 14 hours trying to just get the pipeline to work on their computer. And then even if you get it working on one computer, you now have to set up the pipeline on every other computer at the lab and then train every person to use the pipeline, right? So what we've been doing with the bioinformaticians is giving them an environment to write pipelines, publish them on our platform, which is hosted on cloud computers. Every time they publish the pipeline, it automatically generates a no-code scientific user interface for that pipeline, right? So the liquid biopsy cancer detection pipeline, for example, you have a bioinformatician at a company that writes that and they upload it to Latch. Now they have this UI associated with the pipeline where anyone at their company can go in, drag in their data, and run that workflow. And there's one copy of the workflow, and it has a version associated with it. And it's running on a set of computers on the cloud. So you don't need to install it on your computer. You don't need to do the setup. And don't get me wrong, it is not a solved problem. We're going to be spending 5, 10 plus years like solving this. But I think it is a step in the right direction of trying to create an environment where these things get standardized and they get reproducible. Yeah.
So I have a couple of other, I mean, since you can go, extremely deep in terms of the technology, I have a couple of other deeper technology questions for you. So some of these companies that I've worked with, at a moment's notice, they need, more compute power to be able to run their diagnostics. Is Latch able to flex to be able to meet the needs of clients for that?
It's such a good question. I mean, so much of the reason we exist is because there are very, very large computers that are needed to execute these workflows. So I credit Kenny, our CTO, who is just a brilliant background in cloud infrastructure engineering. And basically, the answer is yes, and I'll kind of talk through how. But we are built on top of a workflow execution engine called Flight, which is basically, it schedules all these compute jobs on different open computers on a cloud provider like Amazon Web Services. So it's built on top of Kubernetes, which is a workflow orchestration system that anytime you click run a workflow, it's actually going, so it's Dockerizing the code in that workflow. Docker's this technology to like just wrap some code and make it reproducible in any environment. It's finding an available computer on AWS, and then it's executing that code on that computer. And these computers can scale up from 3 CPUs and a couple gigabytes of memory all the way up to like 50 CPUs, multiple GPUs, hundreds of gigabytes of memory. And the way you do that on Latch is when you define the workflow in code, you can assign the amount of CPUs, GPU, and RAM you want for each workflow. It is such an awesome engineering effort. Actually, we've taken a lot of inspiration from companies like Google and Lyft and Spotify. A lot of their technology is built on Flight. And it's a big engineering effort that's really been pioneered by Kenny and Max and some of the great engineers on our team. So that was a long answer, but basically, yes, you can do that. It's not perfect, but we're working on making it more flexible compute every month.
I mean, it is something that most companies I don't think through, until you need it. And whenever you need it, it's almost like, what do you mean I can't have an answer for, a week or 2 weeks or whatever, because you're going to put in your data and you've put in so much that, now it's going to sit there and run for however long. But I mean, the great sort of, solution that I've seen from, other smart scientists and smart biostatisticians that I've worked with is, hey, look, contact Amazon and we'll get more compute power and we'll have an answer tomorrow morning instead of, a week from now, which, sometimes those times matter, because if they're working on let's just say a personalized diagnostic and a drug that is expected to launch at a specific date, you're trying to match those two timelines, as the example. And, in this case, it could mean disaster for the overall project if, hey, look, the one thing that's going to hold it back is the diagnostic that it will tell you whether or not if you need this drug or not. So, yeah, really critical.
Yeah, I couldn't agree more. I'll just add that the speed of compute and the cost of compute are both very big considerations. And as an engineer, you have to make trade-offs between the two. The problem with setting up your own compute instances on AWS is that you have to have an engineer at your biotech company spending hours of their time spinning up and then tearing down the compute instances and thinking about the trade-offs they need to make between using this amount of compute versus that amount. I mean, we've seen biotechs with AWS bills in the amount of hundreds of thousands per month, just from the amount of data they're processing. And with something like Latch, the compute is actually only being charged while the workflow is executing. So when you execute the workflow, you're only being charged while it's running, not having to spin up or tear down the instances. So it's like trying to make that as fast of an execution as possible, because that is the impact, right? The scientists are just going to have to wait otherwise.
Right.
And sometimes they might wait hours, but sometimes it might be days or weeks if it's a very big data set, just to get the results back. So, yeah.
Yeah, so I mean that to me in a lot of cases kind of spells the more complex side of this. Let's go back to the lower end of things. I mean, so how is Latch empowering like bench scientists and maybe smaller companies to be able to use your platform?
Yeah. So from the bench scientist perspective, one of the things that makes me the happiest is that a lot of our tools are open and free for academic community. And we have a lot of developers that publish these tools open and free for the academic community. So the flow for them, the impact is basically this. You have before and after, right? Before, the bench scientist runs a sequencing experiment, they get their data back, and they send it off to the bioinformatician. And they wait days, weeks, in some cases, months. We've seen like 6 or 9 months, especially in academic settings, to get the final processed results back for that experiment. And it's just because there's so much of a backlog on that bioinformatician's plate, or because there's so much compute needed, you're waiting for the bioinformatician to set up the compute. So now that's the before. The after is that the scientists can log in, they drop their files in, like the RNA sequencing data is a really common application to look at the differences in gene expression between, let's say, a healthy cell and a cell that is found in a tumor. And they can drag those files in, click a few buttons, click run, and then they show up, they come back 2 hours later, and they have these beautiful plots that show them the differences in gene expression in each sample and what pathways those genes are implicated in. And that impact is reducing that wait time from weeks or months down to hours across many, many labs. It's just the start, but that's the hope, is to be able to do this for thousands of labs.
Yeah, I've been in academic centers before where they're like, look, it's critical. We got to publish by blah, blah, blah. And, not exactly sure, what truly drives their timeline other than, maybe they have, funding that, is paying for the research. And so they need the data back. And, so for sure, I mean, they've got to feel an absolute benefit in the fact that, hey, look, you could possibly have your data in a lot shorter time as well. So when you think about the biocomputing revolution, I mean, I stole this off of one of the taglines for Latch. At the very end, it mentions, hey, look, Latch is looking to build and disseminate the data infrastructure for the biocomputing revolution. When you think about what the biocomputing revolution is, what do you think that means?
Sure. I'll start off by saying this term was coined by my co-founder, Kenny. And so I'll channel some of him here. And I think a lot of this has come from hours of conversation about where we think this space is going. But we see the biocomputing revolution as a few changes. One, putting powerful compute and really just way better software tools in the hands of every scientist. What does that actually enable? All this blockage and all this time being wasted right now, if that's freed up, how much more efficient does that make the modern biologist? How much more quickly can they iterate on experiments? How much more quickly can they find the new indication, the new target, the new cure for that specific kind of cancer they're looking at? And you multiply that across thousands of labs, right? So it's really about huge efficiency of software tools and compute for scientists. And then it's also about access to tools. So a lot of these tools and a lot of what's going on in bio is that things are behind walled gardens like we were talking about before the interview started, when these things are open and open source and accessible, you'll have eighth graders, high schoolers, people in their mom's garage, people who are not even admitted to a lab yet, but who are just deeply curious and interested in biology, working on trying to answer questions about biology. So when you have access to tools accessible to millions of people across the world, again, it's like, what can be accomplished with that? I think the same thing will be happening with miniaturization of labs and also improvement of CROs. Is the future biologists will have incredibly good software tools at their hands, and they'll be able to outsource all their experiments, hopefully very, very cheaply. And we saw Ultimate Genomics reduce the cost of sequencing to $100. If that continues to happen, that's like what we see as this revolution. And I think the last thing too is there's a lot more truly exciting things that can be built, new plants, new animals that can be invented, new materials that be created. But it all starts with curing many, many diseases. And so part of this revolution is really trying to empower scientists to cure the most crippling diseases that affect humankind. And I think that starts with the big things like cancer and genetic disease, but it goes all the way to aging. And you'd be surprised, there's a very non-trivial population here in San Francisco that is genuinely trying to address aging from many different angles and trying to cure aging. So
I don't think that's new. I don't think that's new for San Francisco. I think I think they've been working on aging for a while, but yeah, I do. I mean, from a scientific perspective, not from a surgical perspective, that would be the new approach here.
Yeah. I think it's like weirdly people are more confident that it's actually possible. And that confidence seems to be increasing, but we'll see.
So there are 3 questions that I like to ask every guest. Kyle, what inspires you?
I think a lot. I mean, it inspires me. I'm really inspired by what the future could look like and how great things could be. And I'm really optimistic about that. Like at my core, I really am. That we can build a truly exciting, equitable, prosperous future that most people don't think is possible. And so the elements of that are like human race thrives, the human race survives, makes it to many planets, and creates incredibly beautiful new things on those planets. I think human creativity is one of the most amazing things in the world. And so enabling humans to be creative with the mindset of a scientist and the mindset of a biologist, and eliminating those barriers to being creative within scientific domains, I think that's really inspiring to me. And people like Jennifer Doudna, people like George Church, that have made great discoveries in the life sciences, I think those are very big inspirations. Also, the leaders like Elon Musk.
Yeah, I think I think from an overall science perspective, I mean, it's a great time to be alive, and you're right. I mean, human creativity has brought an awful lot to the world as well that is inspiring. Second question, what concerns you?
Yeah, I think some of the things that we should be aware of, I mean, I talked about access to tools, are things like biosecurity and just making sure that there are very, very good process and lab regulations in place to prevent things like a leak of a virus, and to make sure that there's not access to these tools for people who have malintent. Because I think that's something that would be very concerning. Is the scale of biology is both a blessing and a curse. You have self-replicating nanomachines that can, you know, be viral. So we have to be careful about who gets certain things in their hands. So I'd say that's one. I'm also pretty concerned about how quickly AI has grown over the last few months and years. There was just a model called Minerva released that was a language model that was just fed in like a bunch of basically all text from the internet. And then it also learned how to parse equations. And then it actually started solving math equations, which was completely unexpected behavior by the designers of the model. So yeah, I'm a little concerned about how quickly AI is pacing, and just are we aligned in being able to control that and make sure that it's doing good.
Yeah. Last question. What excites you?
Man, I'm really excited by building a world-class team. So right now, what excites me is coming to work every day, working with amazing people, also meeting people like you. I mean, it's
Thank you.
so amazing what you've done here. And I think every time I talk to a new scientist or a new PhD or an MD, uh, I really get excited by their work and just interacting with them. So I'm very much like a people person in that way. And then building a world-class team of the most like polymathic engineers that are obsessed with changing the world, that are super inspired and that are smart and make me a better person every day, and are actually trying to solve problems from a technical standpoint. So being part of I am the team that we've made, I'm like constantly impressed with every day. And I'm just really excited to continue building that and hopefully create a company that is generational and that is remembered for a long time.
And you guys just received your Series A funding, so I would imagine you're probably trying to grow like crazy as well. Is there any sort of shout out that you want to give in terms of, where people can go to learn more about, jobs and other things that are happening at LatchBio?
Yeah, you can just check out our website. It's latch.bio. You go to the About page, you'll see some different headshots than you normally would. And there's a careers button there. You can click on the careers. We're hiring for software engineers, bioinformaticians, and business development folks. So please check us out.
And then also, I just wanted to quickly mention for anybody that's watching this on the podcast website, I'll have all the links as well for Kyle's profile, in addition to their website, so that you can trace things back that way as well, just in case you happen to just catch this whenever it gets released on audio. So Kyle Giffin, thank you so much for being a guest on the Life Science Success Podcast. I greatly appreciate your being here.
Don Davis, thank you so much for having me. Really appreciate it.
All right. Thanks. Thank you for listening to Life Science Success. For complete details about this podcast, including show notes, how to get in touch with guests, and more episodes, please visit www.lifesciencesuccess.com. If there's someone you'd like for us to invite to the show as a guest, please let me know by sending me a message at the podcast website. Please click subscribe on your favorite podcast app, share the podcast, or tell a friend about it. And last but not least, rate the podcast. Thank you again.